Hi, my name is Caterina Strambio de Castilla, and in this talk I'm going to present to you a set of guidelines and tools that were developed for the collection and reporting of microscopy metadata by a consortium of different bioimaging communities, including the Open Microscopy Environment, 4D Nucleum, Bioimaging North America, and QUAREP LIMI. This talk will revolve around three interrelated sets of questions. Why is quality control and documentation important? What do we need to do to ensure appropriate documentation and quality control? And how can we carry out these procedures in practice and without compromising scientific productivity? The first answer to the why question revolves around the organization of data. Can we find our data? Do we know how a specific data set was acquired so that we can compare it with other data we are producing today? I'm sure that all of us have found ourselves in a situation in which we wish we knew the answer to these questions, and instead we had to acquire new images because the relevant information about previously acquired data sets was not available to us. The second set of reasons we will discuss revolve around the relationship between quality control and documentation and key aspects of the scientific method, such as data quality, rigor, and reproducibility. Finally, the third important set of answers to the why questions has to do with the deposition of image data sets on public repositories, which can massively increase the impact of our work and is rapidly becoming more popular. So let's get started and talk about first data organization. I love this image from Francisca Mao, which is available on Zenodo, because it summarizes very well the kind of situation all of us have probably found ourselves in while trying to gather data for a publication. But the key question is, how do we get out of this lost data desert? Clearly, what we need is a map, and I argue here that the map is provided by the management of relevant information about our data, which can get us out from the desert and into this dreamland of well-organized data. The next topic I want to address, while trying to find a reason why quality control and documentation are important for imaging experiment, is the topic of data quality and reproducibility. As we are all likely aware of, alarm bells about the poor quality and reproducibility of preclinical studies have been repeatedly sounded by drug companies attempting to follow up on drug targets. In this study, for example, it was found that only 30% of the results were actually reproducible. More recently, formal studies were conducted by the Organization for Open Science to examine the reproducibility of cancer research. The results were not encouraging, and the documentation of the performance of microscopes was found as a key contributing factor that could improve reproducibility. The last topic I want to discuss is the importance of quality control and documentation to ensure sharing value for imaging data. It is becoming increasingly well documented in publications such as this one that the deposition of data sets along manuscripts on public repositories can significantly advance science and increase the impact of our work. But in order to extract maximal value from the data we publish, we need to make sure that the data is well annotated with all the relevant information that allow others to interpret it, understand it, and reuse it. I hope that at this point I have convinced you about the importance of quality control and documentation of imaging experiment. So it is time now to tackle the what question. The what question is, what is needed to ensure appropriate quality control, documentation, and reporting for imaging experiments? In short, what is needed is a good research data management practice. And this practice revolves around multiple different elements that are described in this circle. And these practices start from standards and quality control to, for example, image processing and analysis pipelines, and the development of communities where we can meet with other users, custodians, and manufacturers of microscopes to develop together the best procedures. It is important to know that we are not alone in this work, and that guidelines and principles that to help us define what are good scientific data management and stewardship practices have been proposed. And these principles are increasingly being used by funders and publishers to evaluate the quality of our data. In this talk, though, we're going to primarily concentrate on the standards and quality control aspects of research data management. And in particular, we're going to discuss about the information that needs to be collected and reported to ensure maximal data quality and reproducibility. So I've been talking a lot about information and the management of information. So what is this information? The first thing that we need to clarify here is that the information that needs to be collected about image data is usually referred to as image metadata. You have definitely heard about the term metadata before, but what does it actually mean? In order to understand what metadata means, we can start from general description of metadata. Metadata can be understood as a treasure map to find a treasure. It's data about data that can be used to organize, find, and understand this data. But what does it mean in practice in our everyday work? To understand that, we can start by looking at a lab notebook. In a lab notebook, whether it is a paper lab notebook or an electronic lab notebook, we can find a mixture of metadata and data. And in particular, what we're interested here is one specific kind of data which cannot be really found in a lab notebook, but often is found in our file system, which is the image data. We know that images are data, but where can we find the metadata about the images? In order to understand this a little bit better, we can take advantage of this image, which was recently featured on the cover of Nature Methods, in which we can see that the image can be best understood by considering information that should be associated with the image that describe the sample preparation, the image acquisition, and the analysis procedures that we utilize to extract quantitative information from the data. But how can extract this information? Can that be done in the real world? So this is now the time to get to the practical aspects of our discussion. So in order to understand a little bit better about image metadata, let's go back to the structure of a typical imaging experiment. A typical imaging experiment is subdivided in sample preparation, image acquisition, and image data analysis. All of these phases of the imaging experiments can be described using specific kinds of metadata. In this talk, we're going to only concentrate on microscopy metadata, which is used to document image acquisition and can be divided into three subsets: microscopy hardware specification, image acquisition setting, and calibration. Recently, ForDN, BINA, OMI, and QUAREP have come together to define a set of microscopy metadata specifications that extend the OMI data model and empower reporting and reproducibility. I don't have really much time to describe with you in detail about MBO, but you can learn more about it in a poster that is presented in this meeting by Matthias Hammer. Rather, here I'm going to concentrate on what needs to be done to bring the NBO specification to life. And what we need is interoperable tools, software tools such as MethodsJ2 and Micro-Meta App, which you can learn more about in a tutorial this afternoon. Here, I will give you just a brief introduction, and I will start with Micro-Meta App. Micro-Meta App can be divided into two sections. In the first section, we will capture the hardware specification of a given microscope to produce a graphical representation such as this one, in which we have different icons representing different components, and we can capture the information about each of the components. These will result in a JSON file that captures the microscope hardware. In the second part of the Micro-Meta App, we can use similar procedures to capture the settings that were used to produce a given image using a given microscope. And as a result of this, we will produce a JSON file that contains settings metadata. At this point, I want to tell you a little bit about what are the practical uses of the microscope hardware JSON file and acquisition setting JSON file. What can you do with them? The first thing you can do is you can import them into MethodsJ2 and automatically produce methods text for scientific publication. You can also import them into Homero to annotate your image data. You can build repositories for microscope hardware JSON files, such as the one that have been developed by 4D Nucleum on the 4DN data portal, so that you can search microscopes and compare microscopes that were used for different experiments and evaluate how such microscopes might impact the results. And finally, you can use them to fulfill the requirements of REMBI to deposit your images on Bioimage Archive. Having discussed about the practicalities of microscopy metadata specifications, I want to switch gear a little bit and tell you about a little bit more of a theoretical aspect. So we definitely do not want to be in situations in which we are producing just another microscopy metadata specifications. The reason why we think we are not in this condition is because we're working in a community. In particular, we are working within QUAREP, which is an initiative that brings together imaging scientists with manufacturers of instruments and standard organization to develop together specifications that meet the needs of the community. As a case in point, we are doing this as we speak with camera manufacturers so that we can develop specifications that meet the need of all members, from the manufacturers to the users. And this is going to be done in a way that doesn't duplicate efforts and in close collaboration with other community efforts. At this point, I want to thank you for listening and to thank all the people that were involved in this work, and in particular Alex Rigano and Joel Ryan, who developed Micro-Meta App and MethodsJ2. Thank you for listening.